Hi everyone, I'm Lanston. Today I would like to give a short presentation for my research. Spherical Histogram Approximation for maximum flow of brane collectivity. So today I will talk about the motivation, background, method, and result of my research. When it comes to motivation, there are some previous study in computational cognitive science for the channel size of perception. For example, we will show an image to a participant and then we will modify the image a bit and to show the participants again and to see how the knowledge perceived by the participants would be modified. By doing this, we can approximate how the channel size of the knowledge perceived by the participants would be. Another form of knowledge representation would be embedding, especially under the context of AI, artificial intelligence, or machine learning. For example, image embedding or word embedding in natural language processing. This is practically proved by the autoencoder in deep learning. The structure would be an encoder, decoder, and then a bottleneck is between. As we know, the physical substrate of our perception is our biological body. For example, our neural activities. So, how would these neural activities refer to the psychological embedding? Would the bottleneck or the maximum flow areas of our neural activities or neural flows refers to the bottleneck of some kind of autoencoder structure or the embedding size? So, we want to determine the maximum flow areas of our neural collection. The good news, there are some very accurate data regarding brane collectivity. Allen Institute have taken some brane collectivity experiments regarding injection of some virus into a mice brain since the last decade. In those experiments, some select fluorescent virus such as rAAV would be injected into a mouse brain, and then the mouse brain would be taken to a scanner. Intensity and density values of the injection would be obtained, and the accuracy is up to microns. The gradient of the intensity and density values would be used to devise a diffusion graph G by using the fast measuring method developed by Savian in 1999. In the last slide, we've mentioned about anterograde and retrograde. They refer to two different directions. Anterograde refers to the direction from source to destination, and retrograde refers to the opposite direction, from destination to the source. The virus used in the injection experiments are anterograde, but in our research, we are interested in the retrograde perspective instead. That is, how would the destination be affected by the source? To do this, we will gather the edges of different experiments to form a combined graph. All those paths would point to a specific destination P. For example, in the middle image, you can see that there are two paths pointing towards P. There would be one path in gamma P1 and one path in gamma P3, so there are four paths in the final gamma P as the combined graph. So in gamma P, there are four paths, and we will focus on one of the paths. For example, we focus on gamma 2. We can see that gamma 2 in fact consists of multiple edges and vertices. Apart from the same destination point P, there are mi number of other vertices. So for each path gamma I, you can view it as a mi plus 1 times 3 matrix, because in a three-dimensional space, each coordinate consists of three numbers. What we would do next is to recenter gamma I to gamma I prime by subtracting P. So now the destination point P would be an origin 000. And we can do spherical coordinate transformation to see how far other coordinates are away from the origin or away from the destination site. The coordinate refers to the radius and the two angles. It is worth to point out that the transformed gamma prime prime I is still a mi plus 1 times 3 matrix. so the total number of dimensions didn't change. The reason of doing this is to select some points to do the spherical histogram. So we will select some specific values of radius to select those points, and we will call those spheres as the walls. In the first graph, there are 5 paths and 2 walls. R0 and R1 refers to the radii of the walls, and we would use them to capture the maximum flow. So we would take the farthest point from each path to get our max I, and we would determine the radii by using some statistic, for example, percentile of those our max I. After we have constructed the wall, we can select points that are closest to the walls of each path to form walls hitting points, and we would use those points for doing spherical histogram. In each point of the spherical coordinate, there are radius, inclination angle theta, as well as the azimuth angle phi. So we will put phi as our x-axis and theta as our y-axis. And then we can do 2-dimensional histogram in this space. Let's look at the result of a destination site P in the LGD. LGD refers to the dorsal part of the littoral geniculate complex. It is a key intermediary point of the visual system. These are the values of the wall radii of the wall 0, wall 1, and wall 2. R2 refers to 2600 microns, and as a comparison, the brink containing cuboid size would be 13,000 microns times 8,000 microns times 11,000 microns. In the graph, the blue dots refers to the wall hitting points. The darker color in the spherical histogram result means that there are more points being captured by those areas. Let's put the paths and the maximum flow area together in the 3D space. The red dot here refers to the destination site in LGD, and the colored area refers to the maximum flow area captured by our algorithm. The lines refers to the paths reconstructed by the fast matching method, and the colored lines, for example the yellow lines, refers to the paths that pass through the yellow area. So we can see that the subregions with densest flows are being captured by our algorithm. Let's look at the result of another destination site P at IG. In fact, IG locates at the central area of the corpus callosum. As corpus callosum is one of the axon's richest region in brain, we would expect that many of the flows should occur in those regions. In our 3D space visualization, we can see that the maximum flow area obtained by our algorithm is roughly located in the corpus callosum, which is in line with our understanding. Finally, we will look at another destination site at MO1, which is the layer 1 of the somatomotor areas, and we can see that the maximum flow is roughly along the cortex. I would also like to show you the result of spherical clustering, which is an alternative of spherical histogram. Because clustering is not a focus of our research, we will just simply apply Euclidean distance to theta phi, and then we obtain the result as below. Regarding the number of clusters being used, we can try different number of clusters until the Silhouette score is maximized. There are several ways to improve the result. First of all, we may merge the largest bin and the second largest bin to form a third bin. We can keep doing this until the next largest bin is not a neighbor of the current largest bin. By doing this, we can gather the large bins that are locally close to each other. And the second improvement is to use a sophisticated function on theta and phi. For example, in the bottom right graph, we can see that the orange dots and the blue dots are separate. But in fact, in the spherical representation, they are labels to each other, so they shouldn't be classified into different clusters. Instead of using vanilla Euclidean distance function, we can use something more sophisticated to reflect the spherical topology. The spherical histogram is in fact a very efficient algorithm to capture the maximum flow area. The level of complexity is only proportional to the number of paths. I believe that there is a strong relationship between the computational cognitive science regarding the channel capacity as well as embedded analysis in machine learning and the maximum flow areas of bioinformatics, and I call it a golden triangle hypothesis among the three fields. This brings me to an end of my presentation. I hope further study on this topic would help revealing more connections among these three areas.